doing the sessions for OChem and also for GenChem. And but actually, biochemistry is my major, which is interesting. And now I'm actually holding the first session of biochemistry. And let me know if you don't see the screen. And again, if you guys have any questions, feel free to contact Chris Slack or email. You can also even DM me on Slack channel. I think you might be able to find me. And you can also email and let me know if you have any questions. And because we don't have a lot of people today, so if you want me to slow down or have any questions, feel free to do so. Otherwise, I'm gonna start. So today we're gonna talk about the biological molecules, which you probably know a number of them at this point. So including the protein, different types of lipids, and different types of sugar, and also the kind of material that has the genetic information, which is the DNA and the RNA. But today we're going to focus on the amino acid and the subsequent kind of composition that makes up a more complex structure into protein. So specifically with protein, I don't know if you guys have been checking out since the AlphaGo, which AlphaGo, remember like last year or like I think a couple of years ago that they were using AlphaGo to play chess game. And now they're actually applying the artificial intelligence on a program called AlphaFold, which also is developed by the Google team. And what AlphaFold does is basically they're using artificial intelligence to kind of simulate and given a kind of primary sequence of the amino acid itself, and then they're kind of wanting to understand whether the artificial intelligence program can predict the 3D structure, so how specifically it's going to be folded into a 3D shape from its primary structure, which is the amino acid sequence. So why is it so difficult that they have to use artificial intelligence? Although it sounds simple, like you're just putting like a string of amino acid together into some sort of structure, but it's actually a lot more complex than just to have the sequence to congesture, because it can fold in many different ways, and depending on how the structure is intimately connecting to the number of functions that it can do. And consider how much interaction and like how much role that these proteins are playing within the biological system. You can imagine that it is very important for its structure to be correct in order to illustrate its function. And you probably know that a lot of times, because of some of the mutation, just by one or two amino acids position, it could change everything, leading to cancer and a lot other kind of undesirable diseases. And here I'm putting the histone right here that is kind of surrounded by the double-stranded DNA. And we're going to come back to that in the end of the sections with some of the questions that we could be able to answer by how all of these comes together so that it can have this function of organizing DNA as chromosome forms. So here the example is histone, as mentioned. So the first thing is talking about the amino acid structure itself. And the whole structure is pretty simple. You have C alpha that is having a hydrogen and also the residue, the side chain group. And then you're also connecting to a carboxylic group and also with the amine. And together it kind of comprised of the NCC kind of sequence. And when it talks about the direction of an amino acid, it's usually starting with the N terminal and ending with the C terminal. So always remember that it is the middle that is the C alpha that is connecting with the side chain. And one of the things that is very important to remember is to not to kind of memorize everything so accurately, but to understand how it works. So by looking at this uh, structure, we want to think more kind of in an organic chemistry way, which refers to thinking the localization of some of these electrons. Where are the lone pairs and the charges? Where is basically electron rich and electronically deprived? So then you kind of predict how does the peptide form. Basically, is will be the amino acid, the amino the lone pair on the amino group that is attacking the carboxylic carbon and allowing that peptide to be formed. And in between that reaction, you're gonna have a release of a water molecule. So if you do not remember some of those things, you can go back to one of the OChem session, which I have kind of go through the formation of peptide bond and how it relates to hydrolysis. Some of the key important information right here is basically that every amino acid is L except cysteine, which is D, because having the sulfur on the side chain, it changes the chirality on the C alpha. So that is very important to remember. And one more thing is that the glycine would be a chiral because remember that chirality could only be assigned when you have a carbon that is connected to four different compartments. So if you have two of the same, it's not gonna be assigning any chirality. And as mentioned, that the peptide formation is a key component because its partial double bond characteristics, allowing some of the rotation, and also it will strain the rotation and the angle that it can form. And but it also gives us some of the partial planar kind of character, and we will see in a little bit of how it's gonna be working in terms of a higher level of structure levels. So if you don't remember some of these very important things, which I have listed right here, because those are not provided on the day of the test, on the day of your MCAT. So it will be good for you to kind of memorize some of these information, including the 3-letter and the 1-letter code, its full name, and again, some of their structures. And remember that as mentioned, structure related to function and structure related to reaction. Basically, structure would be kind of the ways that you can conjecture some of the property of how you assign them into specific group, whether they are polar, non-polar, or acidic or basic. So the primary structure, as mentioned, is basically a chain of amino acid that is going from the N terminal to the C terminal, NCC, 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 and basically that is what is going on right here. So you wouldn't be going from this direction, but this direction from the NCC and NCC. So in terms of the acid and base, one of the very key important thing right here that could be a little bit challenging is to calculate the isoelectric form. And isoelectric form, if you have something that is nonpolar, which comprises of glycine, alanine, um, phenylalanine, and also a number of things that is not having a charged species on the side chain, then it's going to be simple because all you have to do is to just add the amino group of the pKa and also adding the pKa from the carboxylic group and divide it by 2. Because you can imagine that if you pass, what does the pKa really means is basically when it reaches of this pKa1, it is the time when half of those amino group is not charged and half of them is being proteinate. Is basic. Oh well, hold on. It's the pKa2 actually right here. So half of the population of that amino group will be positively charged, and half of them will be uncharged. So at that time point, it's specifically regarding to the charge of this whole population of the amino group, it will be around positive 0.5. So if you do that, if you do the same thing with basically with the ones that is on the pKa1, which is the time that is basically with an overall kind of charge population will be negative 0.5 for the carboxylic group. You can imagine that if you come into the middle point of this 2 pKa, those positive and the negative 0.5 would become 0, and that is the isoelectric point, which considering the entire amino acid, it will be a charge of 0. So what happens when you have side chain? So if it is an acidic amino acid, what I usually do is that I just add the carboxylic side chain with the side chain of that pKa and then divide it by 2. Whereas when it is basic, then I will use the amino group pKa and adding with the side chain and then divide it by 2. And here I'm going to bring you guys to one of the questions. I'm going to give you probably 2-3 minutes to do this one. If you have answer, you can type it in the chat box if you want. And if you're confused of anything, feel free to ask me any question. I'm gonna come back in 2-3 minutes. Alrighty, so I think you guys have respective answers, which is okay. You know, if there's anything that we kind of bump into that's a little bit obstacle, it's just time to kind of remove them so that we can just get our answer correct in the future, right? So, what I usually do in terms of these questions is I usually just take the middle two values if they are an even number of pKa that is presented. Sometimes they will talk about like right here, the only two charged species side chain will only be glutamine and lysine. So uh, besides the one that besides the N terminal and the C terminal, you don't have to worry about all the NC in the middle because they are connected by peptide bond and not charged. But I'm going to go through this one step by step so that everyone could be on the same page. So the first thing to ask ourselves basically is that to think about what happened if the pKa is below the pH. So if we think in the hypothetical situation, when the C-terminal carboxylic group is, for example, the solution is at pH 7, which is kind of neutral. Consider the carboxylic group being at a pKa of 2.36, you can imagine that that proton is likely to be deprotonate because that pKa is below the respective pH. So that means that it will deprotonate. All right. So what happened when the pKa is equal to the pH? So as mentioned previously, it means that half of the species will be deprotonate of will lose their proton basically, and then the other half of the species will be in a protonated form. And well, in the last place where the pKa is greater than the pH, which right here, if we're still using water as an example pH, then if you think about the N terminal in water, it will be protonate. Okay, so after we kind of straight up all these information, let's look at respective pH values. So at the pH values of 2.36, which is the N terminal, no, the C terminal carboxylic group. Remember that when you are specifically already at that pKa level, half of the species will be half of that C-terminal acid, well, C-terminal will be in COO minus form, and then the other half will be in COOH. So that kind of leaves us in a kind of -5 and something for the C terminal. And then for this one, for the lysine, because the pKa is much higher than the pH, so that means that that one will be protonated, so with +1. And this one is also higher, so everyone will kind of, no, actually the glutamine will be in a COOH form, which is not charged, but with the N terminal, we'll add another one. So it ends up to a total of 1.5. So a positive charge of 1.5. And going to the next one, if we repeat the same step, you will probably get like 0.5. And then the next one would be negative 0.5. And then this one would be negative 1.5. And if you remember the previous one that I kind of presented right here, you would know that you would just have to take the middle two values. Oops, my values are wrong right here. Let me correct them. And then you basically, by taking those, and then you kind of adding them together and divided by 2, it should gives you around 6. So Did I calculate it wrong? Let me use my calculator. Let me see. 9.9. I calculated by hand, so maybe I calculated wrong. I think I might be. No, I actually calculated right. How do you guys got your answer? If you don't want to explain, you can let me know if you still have question after the class today. I'll try to help you out in regards to that, if that is what you would prefer. So let me go back to the presentation mode. Okay. So then the secondary structure, it kind of talks about basically is how those alpha helixes and how the beta sheets kind of form. And here is just presenting one of the example of the alpha helix right here. And some of the questions that you might want to ask yourself, if I'm not sure if some of you guys might be watching the recording, some of the questions that you may want to ask yourself, such that you know whether you can kind of play it in a twice speed mode or something, is that whether you know what kind of intermolecular force will stabilize this alpha helix structure, and also with the beta sheet. What does it mean by parallel versus anti-parallel? And what restricted the rotation, which that is something that we just mentioned a little bit earlier, is basically the partial double bond rotation, angular strain and rotational strain, because of that partial kind of planar conformation that it is adopting. So that restricts the rotation, and therefore there's only so little structure as of the alpha helices and the beta sheets that they can conform to. But you don't only find alpha helix and also beta sheet. You find other things too. You find things that connecting two different helices together. So what kind of amino acid does it usually mediate a sharper turn? And what does glycine usually favor of some of these turn, and why? So these are the questions that you might want to kind of go over when you're reviewing some of these materials in the textbook and in doing some of the questions as well. But I will go it one by one right here if you still you haven't got as familiar with those kind of informations. So in terms of the beta sheet, the parallel and anti-parallel is talking about the directionality of the NCC direction. If you find two adjacent chain of amino acid that is going the same kind of direction, where you have the NCC, and no, actually, with specifically with this one that I'm pointing at, you can see that it is actually going from NCC from the right to the left. So for this one, it's going to that way. And if we look at the second row having the N right here, and then, so if you remember, that is the C alpha first, and the C alpha is the one that's connecting to the R group, which is the side chain. So you can determine that this is a NCC that is going from the left to the right, which is in the opposite direction from the one that is directly adjacent to it. So in that kind of conformation, we call it the anti-parallel, because they are not going in the same direction. Whereas, like, let's say if both of them are going in the same direction, from the N amino group and then to the C alpha group, and then to this carboxylic group, if both of them going to the same direction, then they are parallel. And take a look that what kind of holds these two chain of amino acid together. It is that not by the interaction on the R group, not on the side chain. It is actually by the backbone itself. When we talk about backbone, it's usually just referring to the amino group, which either it is the lone pair on that nitrogen or that hydrogen that is connecting to the nitrogen. And that hydrogen could be a hydrogen donor that is interacting with some of the lone pair with the carbonyl oxygen. And that forms a number of hydrogen bonding that allows them to pull them together. And actually, some of these, although you kind of see it as a planar orientation, but actually those R group is coming in and out of that beta sheet. So this is not exactly the way that ideally I wanted to present, but I do want to kind of show you what I mean by the direction. And secondly, for the amino acid, the alpha helix structure, you can also see that the same thing is the same kind of hydrogen bonding is occurring when it is between this little gap that is between the two within the rotation of a cycle that is going from up to down or down to up. So you see that in along that alpha helix structure. And again, you can see that the side chain, which is the R group, is sticking out. And I also showed you right here as an example that all these side chain is actually sticking out. Is it possible that these side chains are interacting with other things? It's very likely that they are interacting with other things. So in terms of what they're interacting, then that is something that we're gonna be considering in the subsequent slide. So here is the summary. What kind of intermolecular force that is stabilizing these secondary structure? Is this a hydrogen bonding along the backbone, not in the side between the side chain? What is restricting the rotation? It is because they are partial double bond that gives them the planar conformation and restricting the rotational and angle that they can adopt. And because of that strain, due to those kind of conformation that is adopted, they can only adopt certain form, which right here is the alpha helix and the beta sheet. What kind of amino acid mediates sharp turn? Think about proline, because proline itself is very unique. It is making it into a 5-member ring structure. That means that it's not going to be a more chain-like structure, more linear fashion. So that means that is going to be mediating a very sharp turn if there is going to be something that is connecting adjacent along right here between two chain of amino acid on a beta sheet. What kind of thing will give flexibility? It will be glycine. Think about how small that hydrogen is on this side chain of glycine. Then you kind of figure out everything. And again, all these you just think from the very basic, which I pointed out in the very early slide, that think about the structure. If you remember the 3-letter code, 1-letter code, you think about their structure, you know their property. When you know their property, you know what kind of things that they can do in terms of proline and glycine. So I hope those will help you with studying. Coming into the tertiary structure. So tertiary structure is basically referring to how some of all these motifs are coming together. Here I'm showing you one of the monomer with hemoglobin with inside that green kind of component as the heme group. And that is actually maintaining some sort of aromaticity and allowing the electron that is mobilized along that pi pi orbital. So if you need to review some of the requirement in terms of Hund's rule for aromaticity, I recommend you to go back to some of the OChem session that discussed that before, and think about, because of that aromaticity, what kind of things, what kind of amino side chain would favor that interaction with the heme group when it is aromatic. Is of course with some sort of other aromatic group, right? So in terms of these things, again, to help you to review is not to strictly memorize, but to ask yourself really what kind of things would stabilize these kind of conformation, having different alpha helices to come together. And secondly, what would disrupt all these interactions, allowing this kind of structure to be unfold? What kind of amino acid, again, is making the sharp turns, and which I talk about in the secondary structure? What kind of ligand are they interacting? What kind of interaction that they need for those side chain to be participating in that interaction with the ligand? And ligand is usually just something that is binding to the protein itself. In this case, it will be the heme group. I'm gonna show you guys something which I have prepared. Let me see. A window? No. Okay, let me see how I could do um, maybe full screen, my entire screen. Um, that's weird. How do I do this? Do I do this? I don't know how. Oh, do you see my screen right now? Do you see my let me see. Right here? No. It's not showing up. Okay, let me try. Do you see my Pimo? Just no, you don't. Oh my god. Okay. That's okay. Let me see. How could I, because I made it. Wait, hold on. Say Google Meet is sharing my screen though. Um, can you let me know if you did see my Pimo? Because I couldn't see my own screen. Well, just in case you guys didn't see, I will screenshot this one and then just pull it to my own slide. Okay, no problem. Okay. Okay. So let me stop. Oh, you no. I mean, do you see my Pimo structures? Because I open Pimo. Yeah, I have the structure on the Pimo, but that's okay. If you didn't see, I would just make a duplicate the slide and then try to put it right there. Yeah, you guys can still see it. Okay, so let me see. Going back to recent. Okay, yeah. So that was something that I was trying to tackle with a little bit earlier before this lesson. So right here, you can see that all the side chain is sticking out in the backbone, and what is, and you can see that those directional is basically referring to the directionality on within those two beta sheet itself. You know, as I mentioned previously, if it goes adjacently but in different direction, one is going from left to the right, the other one is going down here. So that this is an anti-parallel beta sheet. And remember that it is the side chain is sticking in and out of that data sheet. And you do see that right here with some of the side chain that is sticking right out. And what you're seeing in this dotted yellow line is basically some of the hydrogen bonding interaction that is occurring in between each other. So you can see that all over. And that is how this structure is coming together by the tertiary structures. Basically, is within one chain of the primary sequence, they can form multiple secondary structure. Here it is multiple alpha helices. How do they come together? It is by the side chain interaction that is coming together. And side chain interaction involving some of all the different forms of intermolecular force, including nonpolar, nonpolar interaction, London dispersion, basically. Whether it is hydrogen donating, hydrogen accepting, then that will be a hydrogen bonding group. And then if it is aromatic stacking altogether, then that will be another kind of nonpolar interaction. So all these interaction is adding up together to kind of make this conformation really compact and stable. And how do they, if you destabilize all these things and using something to kind of disrupt all these interaction, you're likely to get to denaturation, which we'll talk about later. So then we come to the quaternary structure. So in terms of quaternary structures, you involve with multiple chain. Well, when I say chain, it means that from one N terminal to another to the C terminal. And here it is multiple amino acid chain altogether. And within each chain, we can have multiple secondary structure. And when this secondary structure comes itself together as a monomer, then it forms a tertiary structure. And when these monomers are kind of interacting with each other and coming together, then that is the quaternary structures. Again, some of the questions that we are asking ourselves is what really stabilize these things? What disrupt these interactions? And why do we always care about cysteine? Because cysteine is the only one that can be oxidized to form the disulfide bond to kind of hold all these structure together if they're coming from different amino acid primary sequence. So here it is the hemoglobin itself that I'm showing you, which is two heterodimer. So heterodimer meaning that they have one alpha chain and then they have one that is called beta chain. And since these two chains have different sequence, they are different. And when they're different, we call it hetero. And because they are two dimer, well, two mer together, so although they're different, we call them dimer. So with two heterodimer together, we form an entire hemoglobin structures. So coming back to the original example that I was giving you in terms of histone structure that is surrounded by this double-stranded DNA that is and kind of wrapping it around. What are some of these level of structure information that might tell us to kind of contribute to the interaction with the DNA itself? We think about the primary structure. The primary structure is the sequence. The sequence determine what kind of side chain is available for the interaction with the double-stranded DNA itself. And remember that the double-stranded DNA is backbone, has a lot of phosphor group. And those phosphate group is all the way along this side chain, and they kind of gives a lot of the negative charge. Let me think. POH, yeah. It is the negative charge that is all along this backbone. And what do you need to interact with these phosphate group in the backbone is that you're gonna need some of the positive charge kind of form that intimate interaction, right? So then we can expect that there must be some positively charged, which is the basic residues within a primary structure, which is the amino acid sequence. Secondary structure, we already see that there's a bunch of the alpha helices that is coming together. So that tells a little bit about the secondary structure, the tertiary structure. Tertiary structure, remember that there are some of them that are coming together to form a monomer, and multiple monomer may form something else. And we can kind of see that they have some structural similarity, so they might be more than one monomer together that are forming the entire structure. And what does it interacting with, which I kind of answered. What does it need for the ligand protein interaction? Remember that the double-stranded DNA backbone are kind of charged. We do need some sort of charged species. What would disrupt the ligand protein interaction? Those are all the questions that we need to think about. So in terms of is individual monomer, you can see that we have 4 different types that comes all together to form a tetramer. And this tetramer, which is similar to the hemoglobin ones, where you have 2 tetramer that is coming together to form an octamer. And then this octamer will be then having interaction with the double-stranded DNA, and then all they come together with 2 looping, and then all together, then you kind of stuff it into that nucleus. The last thing that we're thinking about is the denaturation, which is something that I kind of talk about with each of the level. Is basically denaturation is to kind of unwrap the folding itself. And what do you need to kind of unfold? Well, basically to kind of disrupt all those interaction, right? How do you disrupt interaction? Remember from the very distant historical Gen Chem lessons, basically when a bond is formed, energy is kind of released because the bond is formed so that it is at a lower energy level. So to push it, to break that bond, you're gonna need some energy that is taking to break that bond into, well, up a higher energy level and then separate them. So in terms of the disruption of different kind of interaction, then you kind of think about what kind of interaction that is going on, right? Basically, is a lot of the non-covalent bonding, except the disulfide bond, because it is kind of a covalent bond. But how do you disrupt the disulfide bond? Well, because it is an oxidation process that allows the 2 cysteine side chain to undergo oxidation reaction. Then you want it to be reduced in order to separate them, right? So then in the case of separating the 2 sulfide together, then of course you add some reducing reagent. So then you can separate them. So, but in terms of other breaking down all other non-covalent bonding, you can generally use some sort of strong acid or base using some sort of heat. Even like detergent, what does it mean by detergent? Detergent is soap, right? Soap is basically having a bunch of charges, and guess what they disrupt? Is disrupting all the things that is partially charged, and also the hydrogen bonding that they're interacting initially. So in terms of some of these questions that we would think about is, when it is denatured, does it still maintain a function? I think that sometimes we kind of get confused that maybe they still have some sort of ability to bind because they still have those side chain. But to think about that, you need to think about, uh, for example, let's say this one is the hemoglobin itself. You're gonna need 4 different monomer together to comes together, and then you allow the kind of iron to come to bind, right? If they're kind of in this squiggly pasta shape, do you think that they are able to hold that iron securely? And in terms of what level of structures that is still maintained at this point when you apply the acid and base or detergent or some level of heat, what kind of structure would still be maintained? You see that right here is showing that it is in a squiggly string. And what does it mean is that the peptide bond, although a non-covalent interaction is kind of being disrupted, the covalent one seems to be able to be retained, right? Unless you're putting it into, like, I don't know, laser, a lot more heat, then like this covalent bonding is still likely to be maintained. With this specifically is the peptide bond itself. But if the passage is telling you that the hydrolysis is going to be taking place, what does it mean by hydrolysis? And how does it affect the peptide bond? Remember that when you form the peptide bond, you release a water molecule. So how do you reverse that process? And what does it mean by hydrolysis itself? So think about that when the passage is giving you specific information that you know that is relating to some sort of structure that is initially required to form the entire peptide. And at last, what kind of principle does SDS page utilize to measure peptide size, and at what kind of structural level? So SDS is a detergent, and therefore, what kind of interaction will be disrupted? And I think I have been trying to allude you guys to get to those answer with my own effort. But if you still confused, definitely let me know. Um, and what kind of peptide size does it measure? Does it measure the one that is on the left when it is all folded, or does it measure the one that is like unfolded already? What kind of structural level? So remember all the structural level that we have been already discussed. What is considered as having the 3D shape, and how in terms of the primary structure, that is more of a kind of one-dimensional version. So think about all those questions. So I'm gonna bring you guys to one of the practice of today. Oops, I think I'm running out of time, but I think we'll go over all the questions. Let's see. Uh, I might, since this passage should be, let me see. Yeah, I'm gonna give you guys up to probably 9 o'clock, and then we can starting to go over the questions. Do let me know if you have any other questions. In You can type it out or let me know. Either way is fine. Okay, I will see you guys in a little bit. Okay. Um, I don't know if everyone is ready. But let me see. Okay, you guys ready? I'm gonna start going through the passage itself. Um, so the first paragraph basically talks about what's butyrate, right? And the important things that is noted right here that I would kind of go through it really quickly. That the first line is basically talking about the definition of butyrate in specifically in the study, and then, for example, blah 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 blah. That is to supplement the first sentence, and then right here I underline highlighting the function of butyrate itself. So basically, to kind of the butyrate inhibit this HDAC, and HDAC basically do the opposite things with the HATs, and HATs and that HDAC basically control the chromatin structure within the histone and non-histone protein, and they can only modify basic residue. And then the next thing is that butyrate actually structurally related to da 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 da, and then they used the major source for prolonged exercise and starvation. Okay, and then they say that to determine whether that beta OHB also have this HDAC inhibiting productivity, which is similar to the butyrate above. They are treating with different concentration of beta OHB, and beta OHB, how to measure its activity is to look at the acetylation of specific position on the histone, including this acetylation of the lysine number 9 and also this lysine number 14. And also they are measuring this acetylase on the acetylation on the butyrate, which is interesting. So then the next thing, they show you the graph, they show you a little bit of how the trend goes as they increase the concentration of beta OHB. And then they are also identifying the beta OHB protection against oxidative stress, and which results in carbonyl derivative. So if there's oxidative stress, basically you will get carbonyl that is directed by a metal catalyzed oxidative attack on the amino acid side chain, and then this example, and then you can inject them with the paraquat, which is the reactive oxygen species, and you have that, then you will have the carbonyl derivatives. And if you have the beta OHB, the researcher hypothesizing that if you have this beta OHB protection, then you basically should stop this oxidative stress. If you stop this oxidative stress from the paraquat, you will not get the carbonyl derivatives. And these, how do they measure? They measure by isolating the kidney sample and then assay the carbonylation on the immunoblock. And the result is that the carbonylation is suppressed when they receive the beta OHB, confirming the researcher hypothesis that the beta OHB has a protection against the oxidative stress. So that is the passage itself. So the first question concerned about what is the structure of the amino acid that can be modified by HDAC. We're going back to the paragraph where HDAC is mentioned, which is the paragraph 1. The paragraph 1 saying that it inhibits the HDAC and counters the HAT. And what's the next line saying? Both of them modify basic residue. So that will be our target in the answer choices. So what does basic residue looks like? Basic residue, when they lose, well, no, they pick up an extra proton, right? But basic residue, they pick up an extra proton on the side chain. So we need to look for the side chain. that has an extra proton, and ideally making it positive, right? So the only answer that is suitable for our choices will be the answer D, which is, I believe, let me do some border. Yeah, it is the only one that has the positive charge. Okay, so there we go. And then the next question. So HDAT, how does HDAT change chromatin? Let me copy one of this. Okay. HDAT, they change the chromatin by da da da. So let's think about HDAT. HDAT basically is gonna be acetylase, right? The acetylation, acetylation. Okay, the acetylation, and the acetylation on the amino group. So the amino group that originally is gonna be looking like NH, and then with an acidyl group, will be removing that acetylation so that they will return to the free amino group, like the one that we just saw in the previous question. So originally, you can attach a still acetylation, so removing that positive charge, because once you acetylize it, you do not have the charge, right? So remember what I mentioned earlier when I look at the histone structure interacting with the double-stranded DNA. The double-stranded DNA backbone is double-stranded DNA backbone is negatively charged because of the phosphate groups. Okay. And with the HDAT removing the acetylase group, so making those basic residue become charged. So HDAT basically can increase the positive positive charges on the histone. So what happens when you have more positive charge to interact with something that is negatively charged? So it means that it's gonna make histone and this DNA double-stranded DNA interaction increases. So when they have more intimate interaction, that means that that kind of connection will be more difficult to separate, and leaving us to the answer B, which saying that they're increasing the condensation, allowing the histone to be more able to wrap around that double-stranded DNA. And when that interaction is really tight, how can this transcription mechanism enzyme comes along? So that is not possible. So that's why answer B would be our correct answers. Okay, so that is that. So number three talks about if HDAT inhibition by beta OHB is a physiological responses in living animals, the information in the passage indicate it is likely to be occur when. So let's look at when beta OHB first introduced. Beta OHB is introduced in paragraph 2. And immediately after the introduction of beta OHB, they talk about that it is a major source of energy for mammals during prolonged exercise and starvation. So what happens when you have prolonged exercise and starvation? Your glycogen reserve is gonna be depleted. When your glycogen is all depleted, what do you look for energy? You look for fat, right? So let's kind of think about the answer choices. Answer choices say first they're substituting the fatty acid oxidation. That might be the case because in order to mobilize the energy storage within the fat itself, you have to go through the oxidation, which is called the beta oxidation, right? So that is possible. Pentyl phosphate shunt is activated. When do you activate the pentyl phosphate shunt? Is when you're doing something that is related to synthesis. You're synthesizing either something rivals something, or so rivals will be used for basically doing the RNAs or making up the DNA, which occur a lot when we're doing active cell division, right? But I don't think we're talking about cell division right here. So that might not be the case. Remember that 5 is referring to the pentose is referring to a 5-member ring, and it all happens that within a ribosome, our RNA, it is the ribose, and ribose is a 5-member ring, and therefore it is a pentose phosphate shunt. That is what is referring to making more nucleic acid actually. Whether there is increased gluconeogenesis, remember that we already kind of think about when you're going through prolonged exercise, that means that you're using all up your glycogen storage. So glycogen, you need to do glycogenolysis to kind of break down the glycogen, right? And when you do that, along with some of the gluconeogenesis, then you will get more glucose in your blood. But here is not the case because it says that you're already starved for a very long time and it's prolonged exercise. And one more hint that it might be telling you more is that butyrate itself is a carboxylate, a short chain fatty acid, if you think about that, because it is a 4 carbon and then with a COO in the end. So it means that it is related to fat because it is a short chain fatty acid basically. So those are all the hints that I can provide to you off my head. And if that helps. Okay. So question number 4 talks about which wood cannot be classified with any of the amino acid that is mentioned within the passage that is subjected to metal catalyzed carbonylation. And going back to the paragraph, which is the paragraph 3, that talks about the carbonylation, the amino acid of our choices are proline, which is nonpolar, arginine, which is basic, lysine also basic, threonine is polar. So the only answer choice that will fit our criteria is acidic. Okay, so hydrophilic basically is polar, hydrophobic is nonpolar. Cool. And then the next question brings us to which of the following statements is best supported by the data that is using the anti-acetyltubulin, which is the lysine on the tubulin of a lysine position 40 antibody. So the answer choices are the HDAC acetylate many types of the target protein besides histone. Okay. Beta OHB is not a general deacetylase inhibitor. Okay. And then the next one is it presented in the nucleus but not in the cytoplasm. How do you know? Because tubulin is in the cytoplasm and it looks like they're doing something right here. So I would eliminate answer C, and then beta OHB is inhibit many different deacetylases compared to butyrate. So the thing that I think B and D are both possible answer. A, it says that H that acetylate, but okay. Well, we just mentioned that HDAC is a deacetylation kind of enzyme, so I don't think it acetylate many different kinds, so also not A. So remaining answer choices are B and D. And B and D kind of interesting because they are almost speaking about the same thing, because if you think about what does it mean by beta OHB is not a general deacetylase inhibitor. So it's not general. Basically, it is specific, right? So it is specific goes against with it inhibits different. Well, it inhibits different deacetylase compared to butyrate. Well, I would eliminate answer D because there is no information about comparison to butyrate. There's no data that is supporting that butyrate only inhibits one kind of the acetylase. I will look into the paragraph one more time to make sure. Yep. It didn't say what it is, and there's no data to support. So for me, I think I would choose B. And I believe that is the right answer. Let me see. Yeah. So, yep. Okay. So that is the right answer. And let me put it there. Okay. So that is it for today. Let me stop the recording too. Okay. Okay, let me see. The answer to B is because, yeah, so yeah, I'm correct. So it's not D is incorrect because there's no data to suggest that which H that butcher is acting on. Yep, okay. All right. Let me know if you have any questions. I will always be on Slack, and hopefully those materials would help. And good luck with preparing MCAT.